Hello, and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today, we'll be talking to Drs. Anthony Brusa and Melissa Wilson about recovering Palmer Amaranth seeds from manure. Palmer Amaranth is a super pest spreading its way across the states, and when seeds slip into feed, manure can help this pest to spread all the more rapidly. This episode, Anthony and Melissa share their work finding ways to help farmers arrest the pest, even if their livestock have already eaten contaminated feed. We'll talk more about that in a minute, but before we dive in, we want to thank our sponsor for this episode, Manure Group. Manure specializes in robust soil moisture sensing, innovative weather monitoring, cloud data logging, advanced data visualization software, and more. Their well-published scientific instrumentation is used worldwide in universities, research and testing labs, government agencies, agriculture, and industrial applications. Listen to their new podcast, We Measure the World, to hear how innovative researchers leverage environmental data to make our world a better and more sustainable place at metergroup.com/fieldlabearth. I'm your host, Abby Morrison. Let's talk about science. Hi, everyone. Welcome to the show. Today we have Melissa Wilson and Anthony Brusa with us. Melissa is an associate professor and the manure nutrient management specialist at the University of Minnesota with appointments in both research and extension. Her research focuses on filling knowledge gaps about manure nutrient cycling as farming practices, weather, and technologies change. Anthony is a researcher at the University of Minnesota. He completed his master's and PhD in ecology and evolution at Rutgers University, where his work focused on population genetics of invasive plant species. Anthony joined the University of Minnesota in 2018 to address the emerging problem of Palmer Amaranth introductions into the state. Hello, how are you both doing today?
Great, thanks for having us.
Yeah, doing well.
Awesome. We are so excited to have you on the show today. So we are going to be talking about Palmer Amaranth today. So just to get us started, for people who may not be familiar with this plant, can you just tell us all about it, what it looks like, why it is so effective at its pestitude, and yeah, just how it functions as a pest?
Yeah, so Palmer Amaranth is a weed species that's really high impact in agronomic systems, and it is native to the American Southwest as well as northern Mexico. And it has been spreading across the decades, initially into the southeastern United States, where it caused a lot of problems in the Carolinas and Georgia, impacting a lot of, for example, cotton growth. It was a major pest for. And in the last 2 to 3 decades, it's been spreading further into the Midwest, and most recently into the upper Midwest, and even reaching as far north as Canada. So it's a major concern for a lot of people for row crops, particularly corn and soybean. It's been recorded under very severe conditions to have a 91% yield loss in corn and a 79% yield loss in soybean. Beyond that, it also has impact for livestock because Palmer can accumulate very high levels of nitrates in the leaves, which makes it toxic to animals that would eat it.
Wow, that is so bad. It's like such a high yield rate loss. That's crazy. So what does it look like?
So Palmer is it's what we call an herbaceous plant. So it's not a shrub or a tree. It's it's entirely just green tissue. It starts off as a small little rosette on the ground, but it doesn't stay that way for very long. Under good conditions, it can grow 2 to 3 inches per day, and it can get up to 6 to 8 feet tall total. And then once it's mature, the top part of it produces a large amount of seeds, which can be over 250,000 per individual. So this is part of the problem is just once it reaches seed, it produces an absolutely massive amount of seed, and it's very, very difficult to control it once it reaches that point. The seeds themselves are very tiny. They're about 1 millimeter in size. So think about a poppy seed. They're just small little black specks that you could easily overlook. And they have extremely high germination rate, 95%, 98%, depending on the temperature that they're at. So out of that million seeds, most of them will start growing into a new Palmer if given the chance.
Sure. Is this the kind of plant where in some instances it is like useful for anything that humans do, or is it just like, nah, it's bad news kind of wherever it is?
So there are other amaranths. There is one that's actually called grain amaranth, and that is useful as it's an actual grain that's grown in South America. It's widely used by indigenous people. You can actually buy it at like a Trader Joe's or a Whole Foods or something. You can buy amaranth and you can toast it, and it's pretty good. This is not that amaranth, unfortunately. It is not a domesticated species. It is a very high impact weed. It does not have as much impact in its native range, but once it got outside of the native range, it started taking advantage of all the additional resources. There's a lot more moisture available, which was holding it back. And in agronomic fields specifically, there's a lot of nutrients there because farmers fertilize their fields. And pretty much any sort of a weed species, as soon as you give it those resources, it just takes off running and makes the most use of it that it can.
Sure. Awesome. Okay, that's super helpful for just giving us a framework to operate from. Obviously, an important and intense problem to solve. So you guys were basically trying to figure out ways to stop these seeds from spreading, particularly through manure. So, Melissa, could you tell me a little bit about how these seeds spread via that mean, and also what you were hoping to do in the study?
Yeah, so one of the pathways, and there's multiple ways that Palmer Amaranth has been finding its way into the upper Midwest, but one of the pathways is through contaminated feed that is grown further south, and then it is shipped up to the northern regions for livestock feed. So what actually happened in the case of Minnesota, where we are, is that some Palmer Amaranth was detected in a field, and this field had manure on it, and then they figured out that some contaminated feed had been fed to the cattle from which this manure was spread. So that's kind of the impetus that really got us thinking about, all right, so say we have contaminated feed, but we didn't know it was contaminated, and now it's in the manure. How can we potentially treat this going into the future, that sort of thing? But first, we needed to start with detection, because we have genetic tests, and that's something that Anthony has expertise on. But, you can't do genetic testing when it's covered in manure. So how do we get these seeds out of manure so that they can be tested to see if it's even a problem or a suspected problem in the manure that you have? So that's kind of where we're going with this research, is how can we even get it separated out of the manure so that we can do the genetic testing? Sure.
So I have a question here. For the feed, first of all, can you just explain like how the amaranth gets into the feed that is being fed? Like, are these like corn plants? Is this like grassy feed? What are we looking at there to start?
Yeah, in the case of Minnesota, the first one that we found was actually sunflower screenings. So there was, Palmer Amaranth in a sunflower field, and sunflowers can get really tall, but so can Palmer Amaranth. And what happens is, as they are harvesting all of the seeds, the Palmer Amaranth seed gets mixed in with it. So they shell the seeds, get all the meat out, and that's fine, because the Palmer Amaranth kind of seeds just go with the shells then. So these shells are a good feed source for cattle because they still have a lot of protein and that sort of thing. But then they have the Palmer Amaranth kind of mixed in with them. So that's the first case that we know about coming into Minnesota. In nearby Wisconsin, there's also been cotton screenings, which is a similar process. It's just kind of the byproduct of after the cotton is really taken off and harvested for the parts that are needed. The rest of it can be used for animal feed, but there was Palmer Amaranth seed mix in, unfortunately.
Sure.
I think even with hay, we've seen contaminated hay that has Palmer Amaranth in it as well. In fact, we have photos of hay bales with Palmer Amaranth growing out of it, even as it's baled. So yeah, there's definitely a lot of ways that it can come.
Yeah, sure. So then, is there not a way to like test the feed, or is it something where it's, they're so tiny that it's not really effective to do it based on like the sample size that you would have? Is the manure testing like a second line of defense? Like, what's going on with that situation?
Yeah, as far as I know, you could definitely test the feed, and Anthony can talk more about that, but it's one of those things where you might not know that you had the issue. So it's one of those things like, say you get the feed source in and then it's already fed to the animals, and then you get notification like, oh no, there might have been some contamination there. So it's kind of this, the second line of defense. We wanted to have this just in case, but Anthony can probably speak more to testing of feed materials.
So it's a combination of a sampling problem and also a question of what the regulations are, particularly at the state level, because most states are having their own individual rules for how to address the problem, which makes sense because Minnesota is very different from Georgia in terms of the pressure that Palmer is putting on it. So the original introduction into the state was through conservation resource plantings, so native pollinator mixes. So in that case, we're talking about seed that's being planted, in this case for restoration, but we've had a lot of introductions reported that came through contaminated agronomic seed. Then, of course, there's the manure that Melissa talked about, and also the possibility of it being introduced through heavy machinery that wasn't cleaned properly. It can be stuck to the outside. So all of these are potential introduction routes, and if you can find potential Palmer seed, then you can test it and see what it is. The problem is being able to identify it and get the testing done. And with a lot of the ones I'm talking about, any of the agronomic seed, the conservation seed, or for feed, when you have a large amount of it, you're only testing a small subsample. Now, you can sample from the bottom where the smaller things tend to accumulate. You can set up your own testing regime, make sure you get as many representative samples as you can, but in the end, you can still end up missing it. You can't test every single piece of every single part of the seed.
Sure.
The other part of this is also the fact that, as I mentioned, there's different regulations depending on what you're working with and where you are. So any agronomic seed is required in Minnesota to be certified as being Palmer-free. That's not the case in other states, and that's not necessarily the case, as far as I know, it's not the case in feed.
Sure.
Melissa, do you know?
I don't know.
Yeah, I'm not aware of any regulations regarding the feed.
Sure.
So that is sort of the question of where these seeds are coming from, the route that they're coming through, and then when we finally do manage to identify something that might be Palmer, then we can get our tools out there, and then we can get a proper call and start taking action. But it's that initial detection that is the challenge.
Sure. Yeah, I imagine there's like a cost restrictive element to that too, where like some people are like risk cost analysis to test every single thing is just maybe a little high. Is that a factor? I don't want to put words in your mouth.
Oh, yeah, it's definitely a factor. The genetic test is not terribly expensive, but when you have a bunch of samples, it's going to add up. The work that I was doing with developing newer models of genetic tests, we can test 200 potential Palmer seeds in one go. And if one of them is Palmer Amaranth, we can flag it and say that the sample's been contaminated. So you can run 200 seeds in one go. That's a pretty good amount. But if you have a very, very large amount that you're working with, you could be pulling out hundreds of potential contaminants depending on what your sampling regime looks like.
Sure. Yeah. So obviously an interesting problem with many, many facets in it. So you were specifically looking at this manure, though. So tell me how you were trying to test for these tiny little seeds in manure.
Yeah, this is a fun part. One of the things is because the seeds are so small, it does make it difficult. So we basically screened it. That was our big thing. And then the other option was to use a method that is used for getting seeds out of soil to test a weed seed bank that might be in soil, which was a dispersion method where it's essentially you add some chemicals, that help separate out organic materials from inorganic materials like soil. The soil would drop to the bottom, the organic materials would float. And then we took different methods within those. So like with sieving, we're like, could we just dry the manure and then sieve it? Do we have to like rinse it through the sieve? Those sorts of things. And then we also tried like with and without blending, because with manure, you got kind of two things going on. We have different types of storage systems. So there might be a liquid handling system, which means there's low solids in it. There's not a lot of bedding. It's, pretty liquidy, or even from like oatmeal consistency to almost like water. And that's kind of what we're considering as liquid. And then we have manure that has a lot of bedding in it. And that's just the different way that the animals are housed. So with the bedding, it makes it a little more difficult because sometimes the bedding can be really long strips of grass. And then how do you get that through a sieve? So we tried with and without blending to try to see if, again, that helped when we had bedding in the manure.
Yeah, totally. So what did you find then?
Well, in our case, it turns out the dispersion methods where we add the chemicals to separate inorganic and organic materials did not work because seeds are organic and so is most of the manure. So it actually made the seeds clump to the manure even tighter, which is the opposite of what we wanted. So that worked horribly. But the sieving methods worked pretty well. In fact, having some sort of pressure, so rinsing the seeds through the sieve and rinsing the manure through the sieve actually really helped. Where if we put 100 seeds into the manure, we were able to recover 93% of those seeds, so 93 seeds in the end. So overall, it's a pretty good recovery rate for something that's so small, as Anthony mentioned, the size of a poppy seed.
Yeah, I'm curious about just when you're sieving, like what, because there's other things in it that are bigger that will also not make it through. So, I mean, did you encounter like trial and error with sieve size? And, obviously you don't want it so small that it's, so big that it's going to slip through, but was there any complications in there? Or were you just like, nah, we know what size they are. Let's make it pretty much that size.
Yeah, we had to go through kind of a multiple sieving stage process where you do a bigger sieve to get the bigger chunks out. The rinsing helped because that helped then still move the seed off of the bigger chunks. And then we did eventually have a smaller sieve at the bottom that was small enough to catch the seed. But there was definitely still some sorting that had to happen. You weren't able to sieve out just the seed. There's still like lots of little particles that get trapped on there as well. So that was kind of the second step, was then trying to pull out enough seed that we could then send for testing.
Yeah. And one of the strengths about the test that I had developed is that it was a lot less sensitive to these other contaminants that Melissa mentioned. So you don't need to get just the Palmer seed. If you happen to have other detritus, other organic material in there, the new generation of tests is specific enough, and there's 3 different markers that I developed, that we could still get a reliable result on whether or not it was Palmer, even with small amounts of other contaminant.
Sure. How many like rinse or sieve cycles did you find was like the most effective? Because obviously you don't want to be like, you have to go through 7 or like 20 different sieve sizes. So was there like a good balance that you found in that?
Yeah, we tried 2 and 3 different sieves. And the nice thing about the sieves that we used, it because this is just a small, experiment, we're able to stack the sieves. So it was kind of all done all at once instead of like having to go through multiple sieving processes.
Oh, cool.
So overall, our people were able to get it down to probably 5 to 10 minutes with just the sieving portion. And then it did take another, 15 to 20 minutes of actually looking for the seeds in there to test. But it was pretty quick compared to like other traditional methods of detection might be grow out methods where you actually find the seed or you take the manure and you spread it over moist soil. And then you have to wait several weeks to see what germinates and then if you can identify it. So this is certainly much more rapid detection than like a grow out method. And then I think, Anthony, your method, once you're able to get the seeds, doesn't take that long either, right?
Correct. Yeah, it's a modification of PCR. So once we get the sample, if we break up the seed, get a little bit of DNA out of it, And then once we load up our reaction, the reaction itself runs for about 2.5 hours. And at the end of that, we get our call. So combine all that together, half a day. Now, obviously, if a large company's doing it, they're going to be having a large throughput. You're not going to get a response that quickly. But having a collaborator, next building over who could do it same day is always helpful for getting that turnaround.
Yeah, and it's certainly quicker either way. Even if it takes a few days to do this, it's certainly quicker than waiting a few weeks for a grow-out study and then having someone have the expertise to be able to identify Palmer Amaranth seedlings too.
Yeah.
And one of the main issues with Palmer Amaranth specifically was that visually it's very difficult to identify Palmer Amaranth versus other amaranths that are closely related that would not be a high priority weed species. It's particularly when they're very young, they look almost identical, and even adult ones, you really need someone who has a lot of experience with it to be able to get reliable calls. So between that, and then obviously we're working with seeds that have almost no characteristics that are easy to spot, that's why there's such a big focus on doing the genetic approach. In addition to, as you mentioned, it's a lot faster.
Hi, everyone. I hope you're enjoying the show. Interested in learning more? Anthony and Melissa's paper, Comparison of Methods to Recover Amaranth Weed Seeds from Manure, published in Agricultural and Environmental Letters, is always freely available. If you are a certified crop advisor or certified professional soil scientist, you can take a quiz for continuing education units for this episode, which can be found in our show notes or on certifiedcropadvisor.org. Thanks again also to our sponsor, Meter Group. Meter specializes in robust soil moisture sensing, innovative weather monitoring, cloud data logging, advanced data visualization software, and more. Their well-published scientific instrumentation is used worldwide in universities, research and testing labs, government agencies, agriculture, and industrial applications. Listen to their new podcast, We Measure the World, to hear how innovative researchers leverage environmental data to make our world a better and more sustainable place at metergroup.com/FieldLabEarth. Thank you for being our sponsor. Let's get back to the show. Sure. I am just so enamored with your research. I just think it's like such a fascinating problem that it seems like the Palmer Amaranth is just so good at what it does, and just every layer that you guys are considering in being able to tackle this problem from this, holistic view is just amazing. I'm so happy that you guys are here. So I'm going to stop fangirling and continue the interview. Just had to get that out of my system. But obviously, so this is research that is just immediately seems like very practical to farmers. So I would love if you would talk about just how this is going to impact farmers, like what they can do to engage in this, how to implement this on their farms, things like that. When to test, how much to test, all of that kind of thing. So could you talk about that a little bit more?
Yeah, and thank you very much for fangirling over the research. We love it. And that's one of the cool things about my job is that I work in very applied research. So we're constantly trying to figure out, like, what are the issues that farmers are facing? Is there a way that we can figure out how to help? And this Palmer Amaranth thing, it's like, I'm not a weed scientist at all. I never imagined I'd be doing weed science research when I got into the manure field. So it's been a really cool collaboration to work with my colleagues on this. So now getting back to the applied aspect, one of the things we realize is like, all right, we know we can sieve this out. Can we take this to the next step forward? And like, is there a treatment process where we can actually get seeds out of liquid manure? Because that's one of our hardest manures to deal with when weed seeds get into it. And anyways, treat this liquid manure so that it's safe to land apply so that we're not spreading these seeds everywhere. So that's kind of what our next step is. We just received funding to look at commercially available equipment. We actually have what's called screw presses on the market, which are for liquid manures where they separate out the liquids and the solids. And when talking with manufacturers, it sounds like they have screen sizes that match our sieve sizes. So we actually think that we can screen out all of this manure effectively and like over large quantities of manure. Like we have dairies in Minnesota that have a large amount of cattle, 5, 10, 15,000 cattle, and they have these screw press systems installed already on their farms. So it could be something as simple as just changing the screen size to be able to capture these seeds. But we have to do the research to find out if this is the case. You know, these are under much higher pressure than what we were doing in the lab where we're just rinsing seeds through a sieve. So we do have to see if this is actually something that's viable. But that's kind of the next step of where our research is going. As far as what farmers can do now, I don't know if there's anyone actually commercially using our methods yet, but it is something that we're hoping that people can adopt, especially because, like, I know the state of Minnesota is really interested in arresting the pest is the motto, trying to keep Palmer out of the state as long as we can, and then being pretty aggressive when we do find it in the state to remediate that. So being able to use this method to detect manure that might have it in it, and then, you know, going forward, being able to figure out ways to treat the manure for the seed is going to be pretty important.
Sure. And so is this something that, like, when it's implemented, is this going to be something that is tied in with, like, the regulations that Anthony mentioned? Is it just on the farmer to decide, you know, at least in the early stages? I assume if new regulations are made, that would make a difference. But right now, is it kind of just like, if you want to do it, that'd be great? What considerations would a farmer need to think about in implementing this system in regards to testing their feed or the manure after the feed is given? Like, what's going on there?
Yeah, I mean, this is the big question, and it depends on, like, what region you're in. Like, here in the upper Midwest, we don't have that much of it, so it's not really here. So we are thinking about the feed that's being imported versus if you're down south, this is, like, already probably in your field. So, like, that's a different story. So when we're talking about up here where it's not necessarily a problem yet and we're trying to avoid it, farmers will certainly want to think about their feed sources and try to get, you know, trusted feed sources, know what they're getting into. And the thing is, if they have suspected contamination, there are certain things you can do. Like if you have a bedded system, which has a lot of bedding in it, that manure is compostable. So you could potentially compost the manure. And I mean actively compost it, like not just let it sit out in the field for a while. Like actively turning it once a week, making sure it has enough water so that it reaches the temperatures that are needed to kill Palmer is going to be really important. And then you have to do that multiple times with it being mixed, because then you have to get the seeds from the outside into the center so they get heated too. But that's one option for farmers who have a solid system. With the liquid, it's a little harder. They can potentially go through a digestion system, but that's not like, we don't have portable digesters that go from farm to farm. Like that's already, if it's something that's already installed on the farm. There's been some evidence that some seed germination will decrease after it goes through germination, but I don't know if we know that for Palmer Amaranth specifically. And usually that's actually just a numbers game, because even if one Palmer Amaranth seed makes it through and it germinates and then creates that, 250,000 more seeds in that field, it can become a problem pretty quickly. So it's really a numbers game when it comes to Palmer Amaranth. But the other thing is making sure if you do have suspected contamination, spread it on a field where you can monitor it regularly, and then try to get to those plants before they go to seed is important.
As far as the seed passing through animals, there hasn't been much work done on that. There was a little bit of work done on waterfowl, which is obviously very different from a ruminant. But the seed was able to pass through and still be viable. I'd love to see some work done with either cows or with goats. Maybe we can get our University of Minnesota goat team to look at some Palmer for us. I know people love seeing the goats with diapers pictures.
I think there actually has been some research with like red root pigweed, which has a similar seed to Palmer. So not Palmer directly, but kind of similar enough. And you're right. It's the same thing as a numbers game there. Like we see reduction in germination after it goes through ruminants, after it goes through poultry, but it's still like it's not 100% reduction. So again, if you have even one viable seed, that can become problematic, at least for the small hard-coated seeds that we see with other amaranths.
Can you explain what a digester machine is?
Yeah, sure. So one of the it's not new, but it's we're starting to see more of it. A digestion process is where they are generating gas using manure. So they allow it to go anaerobic, and there's certain bacteria in there that will turn the carbon into methane, and then they can take that off. But then because of the acidity and sometimes the heat and things like that in a digester, sometimes that can help decrease the germination of the seeds.
Oh, okay.
It's kind of like a big stomach that turns manure into gas.
Yeah, that's what it sounded like. So that's super cool. I'm learning a lot today. This is wonderful.
Yeah, poop is actually pretty fascinating.
It is. So I think we've already talked a little bit about some of the future research. But are there other areas that either you or others are looking into to stop Palmer amaranth or develop better testing methods, anything like that?
Yeah, so for the broader question of Palmer amaranth research that's going to be ongoing, unfortunately, I'm not involved in that because I've changed over to working with a different species. But my previous team had some excellent colleagues on it that are continuing work with Palmer amaranth. I should mention Todd Gaines over at Colorado State University is doing a bunch of work with herbicide resistance traits. He is actually the individual who found out the mechanism for why Palmer was able to resist glyphosate, also known as Roundup. So that is a very, very important piece of information for understanding what was going on with trying to control Palmer. And he's continuing to work on other herbicide resistance works in Palmer amaranth as well as other weed species. And his team was excellent to work with, and I look forward to seeing the stuff that comes out of their group in the future. And also some of the initial work for our primers and our genetic testing was done by Eric Patterson, who is now at Michigan State University, and he is still involved in weed science. I believe he's still working on Palmer as well. So those two groups are sort of in the forefront of Palmer amaranth at the moment. Beyond that, the test that we developed is commercially available, so it is something that we are offering to private labs if they want to license it and be able to actually offer this as a service. And Colorado State itself is offering it through one of their business units as a service for people who just want to get a single individual sample or a small set of samples tested. You can reach out to them, and they have some excellent tests. I can vouch for the quality of the test. So some good news in terms of control is that the Palmer seeds, even though they're really aggressive at germinating and growing, they have relatively short lifespan. So if Palmer is found in a field and there's actually seed present in the seed bank, the monitoring is about 5 years. After that, almost all of the seed is no longer viable. So the control efforts, once it does end up getting escaped, it does require really aggressive monitoring, but it's not going to be something you're dealing with for decades. After a span of 5 years, then at that point you're basically Palmer-free if you've not had any further emergence. And I mentioned that the herbicide resistance is a really severe problem, but there are other approaches that are being used in order for weed control, and there is a broader idea called integrated pest management, which is taking lots of different techniques and overlapping them to use different approaches. One of the main ones, the one that I'm currently working on, is the use of cover crops. So the idea in this case is that you have something after your corn or soybean is done, you plant something in the fall, you have it over winter, and then in the spring you have it start to emerge. And there's a lot of benefits to that. In addition to just keeping something in the soil so it doesn't erode, you have these plants growing on the soil, and you already have something to compete with Palmer when it's trying to emerge.
Sure.
And this has been used for a lot of different a lot of different species. The one I'm working on currently is pennycress. I'm part of the Forever Green initiative here at University of Minnesota. We have a lot of different species we're looking at. We have not done any work specifically with Palmer to see the impact of these species on it, but it's been used for a lot of other weed species, so there's definitely a lot of potential for non-herbicide-based controls.
Awesome. Yeah, we'll see about getting some resources in our show notes for how to connect with these people. If you want to get in touch. This has been an awesome conversation so far. I have 3 questions left for you. So the first one is, if people want to learn more about this topic, where can they go?
If you're interested about Palmer making its way into the upper Midwest, the Minnesota Department of Agriculture has pretty good resources on their kind of arrest the pest campaign that they're doing, trying to stop it and trying to stop its movement into the area. I think we've probably been one of the more aggressive states in that regard. And again, Anthony can correct me if I'm wrong, but the Minnesota Department of Agriculture has some really good resources. And I'm sure each state probably has resources through extension or through their Department of Agriculture as well.
Yeah, it's definitely, I would say Minnesota has been one of the strongest states in terms of the response to this. We've actually gotten emails from a few different groups that are in states that do not have Palmer, but they are concerned about it spreading, and they're looking to Minnesota and seeing what we're doing and learning from what we do with what works, how to approach the potential introductions. In terms of the sort of this whole broader information about what do you do, where you get this information, how do you respond to things, it really is sort of broken out into, I like to consider it like 3 main groups. You have your groups like the Department of Ag, who do a lot of they do the regulations and the enforcement, and they have a lot of that information, and they put out fact sheets, and you can go to the website, in our case MDA, but every state has a Department of Ag, and they have a lot of information they put out there. Local university extension groups are extremely important. They do a lot of outreach and education events, and they often are just contacts. If you find something weird, like, hey, is this Palmer? Contact somebody from the extension, and they'll be able to just give you some information about how to follow it up. And then, of course, the third part is the people that you've been talking to for about half an hour now, the researchers. We spend a lot of time developing some of these tools and approaches about how to respond to things to make sure that people are better informed so they know what they have to work with.
Sure. Yeah. Palmer Free is the way to be. You guys are awesome. Thank you for all of the work that you do. Whether it's the people that were able to be on the show or your colleagues, they're all awesome. So then next question is, if listeners then want to take that next step and get involved either in the research or just stopping the spread, what can they do?
I would say look out for any sort of events, educational events, field days. As I mentioned, the extension groups are the ones that are usually running those. There may be other groups to be aware of as well. Talk to any of your contractors or consultants. If you're a large farm, you probably have some sort of a contact that can keep you in the loop about things like this. It really does come down to awareness and education. Know about the threat, know what to look for, and then you won't, if you're not sure what to look for, you'll walk right past it in your field, and then next thing you know, you have a problem. But if you take the time to stay alert and just find something that looks out of place, if you whoever it is, regardless if it's livestock or if it's row crops, you know your fields, you know what's supposed to be there, what's normally there. You see something out of place, maybe look into it. See what's going on, and that is probably the best first step I can give you.
Sure. Melissa, did you have anything to add? You don't have to if you don't.
No, Anthony's done a great job of summarizing things. I just reiterate, make sure that you know that there is a problem. That's kind of the first step. And once you do, as he mentioned, if you see something out of place, especially if you see something that doesn't seem to be responding to herbicides, maybe get it looked at.
That's a very good point. I didn't mention it in that much detail, but Palmer Amaranth, one of the reasons it's such a concern is there's a lot of herbicide resistance that's been popping up, particularly to glyphosate, because Roundup is sort of the big name for herbicides for a while, and we're starting to see a lot of resistance to it in Palmer. It's already resistant to a lot of other ones. ALS and PPO inhibitors are basically the most common ones that I see for resistance traits, and Palmer can have multiple resistance traits stacked on top of each other. It's not uncommon to find Palmer that is resistant to 2 or 3 different categories of herbicides, and the worst case scenario, we have had 2 different reports of 5 resistance traits.
Wow. It's it's a problem. This plant is just really good at being alive and going to more places to be alive. Wow. Okay. Sorry. Taming the fangirl. Last question is, what is one fun fact that listeners wouldn't know about you if all they had was your research?
So, one of the things about me that people might not know is that I am actually an artist in my free time. So I do graphite work, I do acrylic and oil painting, I do photography, all sorts of things. And one of the people in the department here came up to me one day and said, "Hey, you're an artist, right?" And that should have been red flags right there. Long story short, I ended up representing the school in the celebrity butter carving contest at the Minnesota State Fair.
Wow! What did you carve?
I carved a small pumpkin out of a block of, like a 1-pound block of butter that they gave me. And all they gave me was a block of butter, like a plastic knife, and some string.
Wow. That sounds really hard.
Yeah, that's awesome.
It was a very interesting experience. Particularly, sculpture is hard enough as it is, but then you're working with something that melts and gets really, really slippery.
Yeah. Wow. That's amazing.
But it was a lot of fun, even though it's definitely not my field. It was worth doing, and I did not win.
It's a tough world, tough medium. Melissa, what about you?
Yeah, one of the things that people might not know about me is I was actually on TV. I was on ABC's To Tell the Truth as the manure specialist.
Wow.
So that was cool. Season 6, episode 19. You might find me on there. And it was probably one of the coolest things I've ever done and probably will ever do. So I've peaked in my career now.
Oh, no. Did they think that you were lying?
Some people did, yeah. I actually, spoiler alert, so turn off the episode now if you're going to watch it, but I actually got to go on early, and I actually got to lie and pretend that I was something else first, and then I got to stay on, and after the real person left, there was only 2 of us left, and then they had to decide who was the real manure specialist, and so I actually got picked as the first person who was a pie baker. I guess I look like a pie baker. So I did a good job lying, but then only one person picked me as the manure expert. But I like to think that's a good challenge as well, because I had to do the training of the other of the actress who was also being the manure expert. So I did such a good job that they picked her as the manure expert. So I think that's good for me as well.
Wow. That's like extension on such a high level. That's so cool.
I think there was because there's tracking systems, it said there was over 1 million viewers. So that was actually pretty cool.
That's amazing. Well, congratulations.
Yeah, it was cool.
Awesome. Well, thank you so much for taking time out of your days to talk to me. This has been a wonderful conversation. So thanks for everything that you do and for being on the show.
Yeah, thanks so much for having us.
Yeah, thanks for having us on.
Hi, everyone, and welcome to our Student Spotlight, where we highlight the work of graduate and undergraduate society student members. Today, we'll be talking to Carl Kunze. Welcome to the show. Can you start us off by introducing yourself and where you're studying?
Yes, thank you for having me. So, my name's Carl Kunze. I study at Cornell University in Ithaca, New York. And I'm also the current student board representative for the Crop Science Society of Americas until the end of this year.
Awesome. And what are you currently researching?
I currently just finished researching, just to end at the end of April, on 2 research projects. One is developing organic naked malting barley for the United States and New York, and the second project has been developing a winter malting barley variety and research associated with that for the New York State environment.
Great. And if you could have your dream research project, what would that be?
I would honestly say it'd be something along the lines of what I've researched for my dissertation and my previous research, because it's been a very fulfilling project to not only work on malting barley, where you are more concerned just about yield, you're concerned about what does the maltster need, what does the farmer need, what does the brewer need, and then what does the consumer prefer. And a lot of this locally produced barley in New York has been driven by the consumer's demand for more locally produced products in a specific environment. And so if I can work on a crop that's associated with those goals in my career in the future, that would be excellent. And so I would say so far I've had it pretty good, and I hopefully continue to do so.
Awesome. Well, if you'd like to get in touch with Carl about his work, we'll have his contact information in our show notes. Thank you so much for being on the show today, and best wishes on your future studies and career. Thank you.
Thank you.
Thank you for listening to Field Lab Earth. More information can be found in the description below. Thank you.